Welcome back everyone. I think we are ready to start. So we are gonna, so far we learned about different bioreactor option that we have for wastewater treatment, and then we learned about microbial kinetics, different kinetic equation and different kinetic parameters. Okay, so that is actually related to growth, yield and decay of biomass. And so now we are going to jump into the third topic for today. So third topic for today, we are going to learn about different important process parameter for activated sludge process, and then we want to actually come up with some design equation, okay, that can combine both kinetics and process parameters. Okay, so that's the third topic. So what are the important process parameter in activated sludge process? Okay, so process parameter that we can control, okay, to actually meet the regulatory guideline. Okay, so to meet let's say 25 milligram per liter BOD or something, right? That's that we want to achieve. So one important parameter is organic loading rate. So microbes, okay, so you remember when I was actually discussing the batch microbial growth curve, okay, so we mentioned that we need a balance between microbes and substrate, right? We need to make a balance for continuous process. So actually microbes, they also have a kinetic limitation. They also have actually, depending on their growth kinetics, they have also limit, how much, for a certain amount of microbes, okay, they can only treat certain amount of mass of organics. Okay, so that's why one of the important thing is organic loading rate. It is very important how much organic actually you are loading into the system. Okay, so because overloading can actually negatively impact the microbes. Okay, so this is something like, maybe you can eat one plate of rice, but I'm really pushing you to eat 10 plates of rice. Okay, so that means I'm overloading you, right? Or maybe you can solve one problem within 10 minutes. I'm asking you to do 5, right? So that's overloading. So that's why we want to make sure we have a balance. So that's why we need to look into organic loading rate. Okay, residence time. Microbes, they need specific amount of time because they want to grow. If you don't provide them enough time, okay, they are going to leave the system before they can grow, right? So we need to provide them enough time. There should be enough time microbe can be in contact with the contaminants and they can actually remove them, right? So the residence time, this is another important thing. MLVSS, this is another term called mixed liquor volatile suspended solid. So what is mixed liquor volatile suspended solid?
This MLVSS is related to substrate, right?
No, MLVSS is related to biomass. Okay, so we want to actually keep a certain concentration of biomass in the reactor, right? Because we want to make sure that we have enough of biomass concentration to make sure that we can achieve our goal. So what is our goal? We want to achieve let's say 25 milligram per liter BOD. Okay, just an example. So mixed liquor volatile suspended solid is used to measure, okay, the biomass concentration in the reactor. But again, that not only active biomass, there could be some, practically there could be other stuff in there, decay, dead cell of biomass. Yeah.
So how to measure this MLVSS?
You can.
MLSS I know how to measure. MLSS I know how to measure.
So VSS, okay. MLSS is total, right? Total suspended solid. So
we take in the measuring cylinder and we allow it to settle for 30 minutes. Then we know the sludge index something.
No, it's actually sludge volume index.
Yeah,
okay, no, this is not. It's kind of more for settleability. But mixed liquor suspended volatile, it's actually thermogravimetric method, the one that we discussed yesterday.
So we have to collect the sludge and we have to put it at 550.
Yeah, we have to filter the sample. We have to keep in oven at 105 degrees Celsius. Okay, take out. So what we get is TSS. Then we put it back at muffle furnace at 550 degrees Celsius for about 5 to 20 minutes. Okay, then take it out. What left is actually ash or fixed suspended solid. So from the difference you can actually get volatile suspended solid. So mixed liquor, why we call it mixed liquor? Because in aeration tank it's mixed with other stuff, other suspended stuff. That's why we use that term called mixed liquor. Okay, mixed liquor volatile suspended solid. The major portion of that mixed liquor volatile suspended solid is active biomass, but as I said, there could be some other portion, like there could be dead biomass, there could be other suspended solid that is coming from wastewater. Okay, so we need to know how much biomass concentration is there. Yes, please.
What does the activated mean?
Activated mean. So activated is used for the term like microbes, they are activated with oxygen. So we have active microbes in the system. So I'm not sure, I did not name that process. I cannot tell, but my understanding it's activated. So they are active.
Can I answer?
Yes.
So basically the bacteria is live here.
Yeah,
Means
they are active.
They are active in nature.
Yeah.
They are not dead bacteria. That's why it's activated.
Yeah. So they are active biomass or active microbes in there that can treat. So that's how then, I hope that's how they named it. Yeah. So, and another thing is food to microorganism ratio, but food to microorganism ratio is almost similar as organic loading rate. So it's kind of food to microorganism ratio, it's kind of like how to balance between substrate BOD with microbial concentration.
Like stoichiometry.
Huh?
Like stoichiometry.
Yeah, you can tell that, yes. So if this is kind of same as organic loading rate. So if you run the system at very high food to microorganism ratio, then microbe may not perform very well. Okay, because microbes, they have a kinetic limitation. So we should not actually. So those two terms, they are kind of like quite correlated. Okay. So now let's see one by one. So volumetric organic loading rate is the mass load of BOD, okay, per volume of reactor per unit time. So how we can estimate that? We can use this equation. VL, Q is the flow rate of wastewater, and S naught is the BOD concentration in your wastewater, okay, and V is the volume of the reactor. So if from the unit you can see it's kilogram per meter cube per day. So this is kind of kilogram of BOD actually. So this is how much, like what is the mass load of organic in your system per day, okay, organic loading rate. And typical range of volumetric organic loading rate for activated sludge process is 0.5 to 1.5 kilogram BOD per meter cube per day. So what would happen like if it's more than 1.5? Any thought? Efficiency will decrease, right? Because they have some limitation, but you want them to actually work more, right? So efficiency will decrease. So sometimes it's better to actually, like, let's say if your process is not performing well, it's better to actually do a simple calculation and see whether you are overloading the organic, like overloading the system or not. So residence time, there are two types of residence time actually used, okay. So residence times for wastewater and residence times for your biomass or microbes, okay. So for
HRT and residence time is the same thing, right?
yes, so there are two types of residence time. One is hydraulic residence time, which is for wastewater, and there is a residence time for solid residence time, okay, which is SRT. Okay, SRT is more for biomass, residence time of biomass in the system. Okay, so HRT basically indicate average time of wastewater remains in the bioreactor in contact with microbes, while SRT indicate the time of biomass or microbes or solid remain in the reactor. Okay, so to achieve a given degree of treatment, so we have to make sure that we have a minimum, like residence time is at least provided to make sure that microbes, they can treat the wastewater, okay, and at the same time they can grow. So when we have, so HRT is the average length of time wastewater actually remain in the reactor, and how we can actually calculate that HRT. So one of the thing I just want to mention, because we want to use that a lot in our kinetic equation. So for HRT we are going to use theta, and SRT we are going to use theta c. So HRT theta and SRT c. Okay. So HRT is basically simply can be calculated volume of reactor divided by the influent flow rate. Okay. So if you know your wastewater flow rate and if you know the volume of your reactor, you can divide the volume by the flow rate and you can simply calculate HRT. But for SRT, it's kind of the residence time for solid. Okay. So you have a reactor, you have certain biomass concentration, but if you remember activated sludge process, we have a settling tank, we are recycling some biomass, but at the same time every day we are taking some sludge out from the system, right? So we have mass of solid in the bioreactor and solid wastage rate. So if I divide the mass of solid in the bioreactor by the solid waste rate, that can give us solid residence time in the system. And solid residence time, some books also use other terms, sludge age, mean cell residence time, okay, also used, but SRT is kind of like, I think, very easy to use. So we are going to use SRT. So how to calculate HRT and SRT for when we have a simple CSTR reactor where we don't have any settling tank that we can actually recycle biomass. Okay. So let's say this is a CSTR and we have wastewater flow rate Q and volume of the reactor is V, and our biomass concentration there is X. Okay. And for CSTR reactor, volume going in, okay, flow rate in and out is gonna be same, and it's completely mixed system. So biomass concentration inside the reactor and in the effluent gonna be the same, right? So X biomass concentration and in the effluent it's gonna be the same. So now if I look into the equation for SRT, mass of biomass in the bioreactor. So mass of biomass is volume multiplied by concentration. So Vx, and biomass or discharge rate, you have effluent flow rate is Q and your concentration is X, right? So divide by Qx, but X we can get rid of it, and so V over Q is, so it's same as HRT. So when we have a CSTR, our SRT and HRT is same, right?
Means if in activated sludge we don't have the recirculation,
yeah,
it will be the same.
Yeah, SRT and HRT gonna be the same. But why we use a settling tank in activated sludge process? Because we want to decouple that SRT and HRT. We wanna treat wastewater within a shorter time, but we want to make sure that microbes can actually stay in the system for longer time. So we want to maintain shorter HRT but longer SRT, and that's the reason like why we use a settling tank. Okay. So now for activated sludge process, let's say we have wastewater flow rate Q, we have an aeration tank which is completely mixed, we have a secondary clarifier, and the final effluent volume is like flow rate is QE, and in the effluent our biomass concentration is XE, and our discharge rate is QW. So we are taking some sludge out, and sludge concentration, like that settled sludge concentration, obviously this XQ gonna be higher than what you have in the reactor, right? Because this is settled and or thickened sludge. So XU. So if we do if you see the system boundary, so here this is flow rate is going in, this is out, right? And so sludge is leaving the system from 2 points, one is here, one is here, right? Yes, obviously this is treated or supernet, and so maybe gonna be very low concentration, right, of solid or suspended solid. And so we know the equation for SRT. So total mass of biomass in the reactor gonna be V multiplied by X, right? Volume multiplied by concentration, and sludge wastage rate is QW multiplied by XU for 0.1, and for 0.2 it's going to be QE multiplied by XE. So our equation for activated sludge process, SRT equation, going to be Vx divided by QE multiplied by XE plus QW multiplied by XW, right? So let's write it down. So HRT, so theta V over Q, SRT theta C Vx QE XE QW, right? So this is the equation. Everyone can read my handwriting, okay? So so residence time, the selection of residence time is very critical, again, because if we don't provide them enough time, microbes, they are going to be washed out, they cannot grow in the system. And typical HRT for activated sludge process is 4 to 8 hours, okay? But on the other hand, SRT is 3 to 15 days. So activated sludge process allow us to decouple SRT and HRT. We maintain shorter HRT but longer SRT. So let's see some simple calculation, okay? Calculation how we can calculate SRT and HRT. So a wastewater is treated by an activated sludge process. Determine the SRT for the following operating conditions. So we have a flow rate of 10,000 meter cube per day. So suspended solid concentration in the effluent is 10 milligram per liter. Sludge wastage rate is 100 meter cube per day. Volume of aeration tank is 5,000 meter cube. Concentration of biomass in aeration tank is 2000 milligram per liter, and concentration of microbes in the effluent from the secondary settling tank, settled sludge is 12,000 milligram per liter. So calculate SRT and HRT. So if we see, we have a flow rate Q, we have XC, we have QW, V, X and XU, right? So we have almost everything that we need for SRT calculation and HRT calculation. So yeah, I just put all the information that we have in the equation, and HRT V over Q, and SRT we can simply use this equation. So SRT gonna be 7.69 day, right? And now the second example, calculation of SRT. So I was actually in the beginning of today's lecture, I was mentioning that instead of secondary settling tank, another option we can have is membrane, right? We can have membrane separation. So this example is showing membrane separation. So an aeration tank followed by a membrane biomass separation until will receive a wastewater flow rate of 35,000 meter cube per day. Calculate daily sludge wastage rate when system will be operated an SRT of 50 days. Okay, assume an aeration tank volume of 500 meter cube and biomass concentration is 2500 milligram per liter. So here, if we see this equation, okay, what would be this concentration? 0, right? Because membrane gonna separate all the biomass, okay? They are gonna be, so only the sludge gonna be wasting from only one point, and one point gonna be here, right? So now let's see how we can calculate that. So I put all the information there. So solid waste, so this is actually what we want to calculate. So if I divide the mass of solid in the bioreactor by SRT, I can calculate the solid or wastage rate, right, in the system. So we know the volume, we know the concentration of biomass, we know the SRT, right, 50 days. So typically for membrane, SRT is really high, okay. So typically for membrane bioreactor, SRT is really high. So sludge wastage rate gonna be 25 kilogram per day, okay. So another term I mentioned that, like organic loading rate. we sometimes you may also actually calculate food to microorganism ratio for the process, okay, to see whether you are overloading the system or not. And how we can calculate food to microorganism ratio? We can, so this is basically balance between substrate and biomass, right? So how we can calculate the mass of substrate in the system? So it's going to be your flow rate, okay, and they are going to be your substrate in for a certain concentration, they are going to be substrate out, right? So from the difference we can calculate, like, you know, what would be the mass of substrate inside the system, right? So if you have a process, right, you have BOD in, BOD out, and you have a flow rate of Q. So I can calculate the mass of substrate Q equal to S naught minus S, and mass of biomass is going to be the same, volume multiplied by concentration of biomass in the system. And for conventional activated sludge process, it's recommended that we keep food to microorganism ratio between 0.2 to 0.5, okay. So if it's higher than 0.5, again what you are going to see, you are going to see performance going to actually decrease, okay. So basically you are asking them to do more than they can do, okay. So another term we mentioned, mixed liquor volatile suspended solid. Again this is more for biomass, okay. So this is more for biomass, but why we call it mixed liquor? Because biomass is going to be mixed with other stuff. It's going to be like, you know, dead biomass, there are going to be some volatile suspended solid coming with incoming wastewater. So everything going to be mixed together. So that's why it's called mixed liquor. But the major portion they are going to be active biomass, okay. And typical range for MLSS in aeration tank is 2000 to 4000 milligram per liter, okay. Active biomass concentration you have to maintain in the system. And again some other, so when we talk about kinetic parameter, we know that yesterday we learned that actually the degradation rate changes with temperature, right? So sometimes you might have kinetic information for some microbes at a certain temperature, okay. So let's say you did some laboratory experiment, you come up with some kinetic coefficient, but you did your experiment at 20 degrees Celsius, right? But now let's say you want to design something at 30 degrees Celsius. What you are going to do? You cannot just directly use them, right? You have to estimate new kinetic parameters considering like the Van Hofer-Henias equation. So like the one that we used yesterday for BOD calculation, the same thing we can actually use here. We can estimate new kinetic parameter at different temperature using Van Hofer-Henias equation. And so lower temperature required because biological process, when temperature is low, microbial growth gonna be low, right? So when growth is low, we need to provide them more time to grow, right? So at low temperature, we need longer residence time, right? And pH in activated sludge process, those are heterotrophic microbes and they prefer neutral pH between 6.5 to 7.5. So typical operating range for conventional activated sludge process is SRT 3 to 15 days, HRT 4 to 8 hours, MLSS concentration 2000. It's kind of like I'm summarizing everything what we learned so far. So MLSS concentration 2000 to 4000 milligram per liter, FM between 0.2 to 0.5, and volumetric BOD loading rate 0.5 to 1.5 kilogram BOD per meter cube per day. So you wanna kind of be within this range, okay? And this is for domestic wastewater, but for industrial wastewater it might be little bit different, right? So now let's do an example, okay? And based on what we have learned so far. So a wastewater is treated by activated sludge process, HRT 7 hours. Calculate the volume of aeration tank, SRT, volumetric loading rate, food to microorganism ratio for the process, considering we have a flow rate of 1800 meter cube per day. influent BOD is 300 milligram per liter, effluent BOD is 25 milligram per liter, sludge wastage rate is 34 meter cube per day, and we have a concentration in the waste sludge 9000 milligram per liter, volume of aeration tank 5000 meter cube, and MLSS concentration, which is X, 2500 milligram per liter. So first HRT calculation. So from HRT we already know 7 hours, but I'm going to convert, it's in hour, I'm going to convert it into day, okay, because everything is in day, like all the kinetic parameter. So volume divided by flow rate is my HRT. So now flow rate multiplied by HRT is my volume. So 0.3 day multiplied by 1800 meter cube per day. So I need a volume of the aeration tank about 540 meter cube. SRT V volume, now we know the volume and we know the concentration of biomass, which is 2500 milligram per liter. So my mass of solid or mass of biomass in my reactor is volume multiplied by concentration. So 540 meter cube multiplied by 2500 milligram per liter, and so solid wastage rate was given 34 meter cube per day. So I divide by 34 meter cube, and so basically flow rate multiplied by concentration 9000 milligram per liter. So my SRT is 4.4 days. Okay. Volumetric organic loading rate is Q S naught, like initial BOD, how much I am loading in the reactor, divide by the volume of the reactor. So 1 kilogram BOD per liter per day. Okay. And FM ratio is Q S naught minus S, right? So we can have the concentration inside the reactor, divide by the volume of reactor, multiply by the concentration of biomass in the reactor, right? So it's 0.37. So can we comment on that? So is it within the recommended range or not? So SRT 3 to 15 days, HRT 4 to 8 hours, FM ratio 0.2 to 0.5, volumetric loading rate 0.5 to 1.5. So if I see SRT 4.4, so it's within the range, right? Volumetric organic loading rate, it's 0.5 to 1.5, right? It's within the range. Food to microorganism ratio, recommendation is 0.2 to 0.5, it's 0.37, so we are good, right? So those are simple calculation, but I'm not doing too much practice problem today because I have to finish by 1:30 because you have a field trip, right? So that's why I'm not, but I would recommend, I think end of the day you are going to have this slide available, okay? Please make sure that, like before you kind of see the solution, try to solve it by yourself and compare your result, okay? So use them as a practice problem. So now the most, I think I would say the most interesting part of today's lecture, which is now we want to develop some equation, okay, that we can actually use as an optimization tool or as a design tool, okay. So if we are actually running some wastewater treatment plant, okay, so and if we want to optimize some process, what we want, always most of the time our goal is we want to meet the regulatory guideline for BOD, right? So what if we have an equation, okay, if we have all the kinetic information, okay, and if we have the operating condition like SRT, one important thing, if we have an equation where we plug the SRT, we plug the, all the kinetic information, value for mu max, value for KD, value for yield, okay, and if we can estimate the effluent concentration, okay, that's going to be like, a very useful tool, right? Instead of doing some experiment, I can put some residence time in there and I can predict, okay, what would be my BOD concentration in the effluent, right? So that could be a very handy tool, and this is actually what we want to do here now. At the same time, we know that actually we need a certain concentration of biomass, right? So what if we have a tool and actually, or we have an equation that can allow us to estimate the biomass concentration, Okay. So if we want to meet a target BOD, okay, so that could be another handy tool, right? So we can maintain a certain, like, we can just check the biomass concentration and make sure that we have enough biomass in the system to treat our wastewater, right? So that could be another thing. We also want to predict sludge wastage rate, okay, but for this one we already have an equation. If we know the SRT, we can just simply use this equation. So we don't need to develop another new equation here. But number 1 and 2 could be a very handy tool for us, okay, and that can be actually used as an optimization tool. Another thing, and engineering, one of the term we always use is safety factor, right? So we know something minimum that we can do and we can achieve our goal, but in engineering we don't do the minimum, okay, we always consider some safety factor in our design, right? So we want to learn about, okay, how we can actually get some insight into the safety factor for biological process considering the kinetic parameters, okay. So how we can actually consider safety factor in our it's kind of like, when some engineer, civil engineer, they design some building, okay, so they predict some, for example, residential building or shopping mall, it's not going to be the same, right? So you have to consider different design safety factor. The same thing for microbial process as well, right? So there should be some safety factor there. If you don't consider safety factor and if you just provide the minimum, at certain point the process may fail, right? So we also want to learn about the design safety factor, or here the safety factor going to be like based on minimum SRT. What is the minimum SRT that we should maintain in our system, okay, to make sure that, like, system not going to fail, okay. So that's actually now what we are going to learn. So we want to get an equation that can allow us to calculate final effluent concentration by manipulating the operating parameter like SRT. We want to calculate what is the MLSS concentration or biomass concentration in the reactor, and we also want to estimate biomass wasting rate, okay, we already have this equation, and at the same time we want to learn about design safety in biological process. Okay, so activated sludge process. So this is kind of like the, the diagram that we used many times, even when I was solving some problem. So how we can actually use this to develop some design equation, okay. So to establish some design equation, we need to use the principle of mass balance and microbial kinetics. So we have to do some mass balance around the system, okay, and we have to use the kinetic equation, okay, in there, and then we can come up with some equation that can be actually used as a design tool, okay. So first thing, we are going to actually do a mass balance for biomass in the system, okay, and first thing we are going to consider steady state. So that means the condition of the system not going to change over time, okay, and there are going to be no accumulation of mass or energy inside the system, right? So but simple mass balance equation is rate of accumulation equal to rate of inflow minus rate of outflow plus net growth, right? So now biomass accumulation in the system, which is we can calculate biomass changes over time dx by dt multiply by the volume, okay, which is accumulation in the system, and rate of inflow, biomass Q is our influent flow rate, right? Q is our influent flow rate, and we consider, okay, in incoming wastewater our biomass concentration is x naught, okay, x naught, and what is outflow? Outflow is from 2 point, okay, here and here. So here outflow is going to be QE multiplied by XE minus QW multiplied by XU, right? You agree with that? Okay.
Can we not add that difference net?
Sorry, what?
Difference net.
Yeah.
Q into x naught plus QR into x. That returns that you are adding the
This is overall
So that
balance.
will be rate of inflow.
No, this is actually we are kind of doing the overall system. Okay, so you have 3 points here. 1 is going in, 2 is going out.
Even inside is also going right. Return sludge also going inside, right?
Return sludge going inside, yes, but this is my system. This dotted line is my system boundary. So I am considering 1 is going in, 2 is going out.
The inside part you are not considering.
No. So I'm actually, so that dotted line is my system boundary.
You can consider like it is circulating
Yeah.
inside the system. You can
How
consider
much,
like
how much amount of return sludge will be taken as a taken to the return aeration tank?
usually 10 to 20%
Of
of
20%
Of the
secondary sludge.
secondary, yeah.
Sludge.
of the second sludge.
Yeah.
Usually, practically.
Yeah, usually, but you have to make sure that you know, like,
That changes normally from system 10 to 20.
okay. So here we are doing the mass balance actually around this system boundary. So we are considering this is going in, this is going out, right? So now the net growth is actually V multiplied by dx by dt net, okay? And now because it's steady state, so there are not going to be any accumulation, okay? Then so this part is going to be 0, and we consider that, okay? So to make our life easier, we consider that there is no active biomass coming from with incoming wastewater, okay? Yes, but there could be some aerobic microbes, you know, in the sewer network, right? And that can actually come with your wastewater. But for modeling purpose, we consider this is kind of 0, okay? So this part also going to be 0. So now if this 2 part is 0, how the equation going to look like? So equation going to look like this, right? So now you can see here, previously I wrote an equation here. Now we are going to use them. So dx by dt net, right? Mu max minus kdx. We already learned previous slide, okay, in one of the previous slide. So now what I can do, I can actually replace that part, right? I can replace that part using this equation. So now, so if we do some rearrangement, and what we are getting here, Vx, Qe, Xe, Qw, Xu, right? And so do you see this? This is actually our SRT, right? So 1 divided by SRT. So now I can actually replace that with 1 divided by theta c, okay? And Kd I can actually take it on the other side, okay? So now my equation look like this. So now what else I can do? If I can, so now my target is, here what you see, you have theta c, which is SRT. What is Kd? Decay rate or decay coefficient, right? And what is mu max? Maximum specific growth rate. What is S? Substrate concentration of what? Like BOD concentration of what? In or out?
That is varying with time.
S naught is in, S is out, right? It's out. So this is my effluent BOD concentration, right? And Ks is what? Half saturation concentration, right? So if you look into this equation, Kd, mu max, Ks, they are kinetic parameter, right? They are fixed for microbes. And what is S? This is my target BOD, okay? Effluent BOD, that's what I am targeting. And what left in this equation? SRT. So that means for a certain microbial community, those kinetic parameters are going to be fixed. So what I can change is my SRT. So by changing SRT, I can get different effluent concentration of substrate, right? So that means if I know the kinetic parameter for a specific microbial community, I can estimate, okay, I can put a target S and I can calculate, okay, how much SRT I need, right? So if I do some rearrangement, this is my equation gonna look like, right? So in mu max, we learned that actually mu max, so K equal to mu max divided by Y, right? So mu max equal to Ky. So I just simply in mu max, I just replace this with Ky, okay? So if you, so now this is our final equation. So this equation can allow us to estimate or predict what would be the effluent concentration from my system, right? And if I do some experiment and estimate kinetic parameter like Ks, Kd, okay, Ks, Kd, yield, right? So only thing I have, I can actually, my process parameter is SRT here, right? So those are biological, for a certain biological population, those kinetic parameters gonna be fixed, right? So the effluent concentration, so which is basically our design goal, okay, as a function of SRT, which is my design variable or operating variable, right? So this design equation can help us to optimize the process, okay? It can help us to find an optimum SRT for my system to meet some discharge or effluent concentration, okay? So now our first objective is done here, okay? So second objective, so for previous one we did mass balance for what? Biomass, right? So now we wanna do what we are gonna do a mass balance for substrate, okay? Gonna do the same like mass balance equation. We are gonna have a rate of accumulation equal to rate of inflow minus rate of outflow plus rate of substrate utilization, right? So now V volume ds by dt and Q is the influent flow rate, and we consider S naught is the initial BOD, okay? And here in the effluent, so from 2 point effluent and sludge wastage rate, okay, BOD concentration gonna be the same, okay? Whatever your, because this is in liquid, right, in dissolved, so whatever effluent concentration or BOD concentration in your final effluent is, it's going to be the same in waste sludge, okay? So QE plus QW, Q plus QW multiplied by S, which is my final BOD, okay? And minus V ds by dt, because substrate going to be utilized, it's going to decrease, so that's why we put a minus sign. So now for steady state, this accumulation going to be 0. Okay, so now this is what we have left. So now if I, so Q S naught minus S divide by V, okay, and mu max X S divide by Y Ks, right? So basically here substrate utilization rate, we have this equation. So what is that equation? This equation, right? So I just replace RSU with this equation here. So now we can do a little bit of rearrangement, and if you can see here HRT, and what I mentioned, HRT we use theta, SRT theta C, right? But what is HRT? HRT is V over Q. And here you can see you have V over Q, right? Q over V. So it's going to be S naught minus S divided by theta. So now you can see we kind of like, you know, like plug those RSU. First we replace RSU with this equation, and then V over Q we replace with HRT. So now, so we can do some further modification of this. So now on the left side you can see mu max S divided by Ks plus S, right? And we kind of found that in one of the previous slide, I think where yeah, this part I forgot to write from previous slide, but here you see mu max S divided by Ks plus S equal to 1 divided by theta c plus Kd, right? So I can simply replace that part here, okay? So this part I can replace with 1 divided by theta c plus Kd, right? So now if you look into this equation, okay, I have initial BOD, I have final BOD, I have yield, I have HRT, I have biomass concentration, I have SRT, I have KD. Here KD Y is kinetic parameter, right? So they are going to be fixed for some microbes. My initial BOD, I am going to know what my characteristics is. Final BOD is my target, what I want to achieve, right? So here if you see what left in this equation is theta, theta c and x. So SRT, HRT and biomass, right? So that means if my other stuff are fixed, okay, I can basically manipulate SRT and HRT and I can maintain a certain biomass concentration, okay. So now if I do some further rearrangement, this is my an equation for x. That equation can help me to calculate, okay, biomass concentration in the system. Typically this is MLVSS, okay, but major portion is active biomass, okay, but practically, okay, that active biomass again and again I am mentioning that, you know, like it's mixed liquor, right? So it's not just active biomass. Biomass is going to be mixed with other suspended solid and you know, like dead biomass, okay.
Now it's clear to me. It is also including the VSS from influent.
Yeah, the VSS from that's why you call it mixed liquor, okay. But this is more for, like, you know, active biomass, but in practically the number might be little bit higher, okay, because of other component. So now we have two equations that we just derived, right? And we have this equation, we already have it from here, sludge wastage rate. So from, we had 4 objective. What was the final objective? We also wanted to see safety factor, right? Safety factor in our design. So critical SRT and design safety factor, this is actually a very important thing when we design any kind of biological process, not necessarily just activated sludge process. So we have to maintain a minimum SRT in the process to make sure that microbes, they have enough time to grow. And why? If microbes cannot grow in the system, okay, so they are going to be washed out from the system, right? So if you don't have microbes, you don't have any treatment, right? So how we can calculate minimum solid residence time, okay, or minimum sludge age or mean cell residence time, okay, for a biological process, okay. So we can estimate that from actually kinetic parameters, okay. So SRT minimum is the minimum point, okay, at which actually biomass is going to leave the system, okay, more rapidly before they can grow. And if they cannot grow, we don't have our treatment, simple thing. So how we can estimate the minimum SRT, okay. So again we have this equation, okay. So we had this equation from one of the previous slide, kinetic equation. So we have SRT, we have mu max, we have S, we have Ks, we have Kd, right. So at critical point, okay, so critical point means no treatment. So no treatment means your BOD in gonna be BOD out, right? No removal of BOD. So S is gonna be same as S naught, right? So S is gonna be the same as S naught. So to calculate minimum SRT, we are gonna assume that S is equal to S naught, and we are gonna plug all the kinetic parameter, okay? And then the value for SRT that we are gonna get, it indicate minimum SRT that we must maintain to actually avoid any biomass washout in the system, okay. So this is minimum SRT, and safety factor is basically design SRT, what we actually use in reality and what is the minimum we need. Okay, so design divide by minimum. Okay, so this is safety factor.
Minimum is kind of theoretical.
Yeah, yeah. So and typically for biological process, safety factor may range from 2 to 20. Okay, or maybe even more than that. Okay, so this equation, it's not necessarily just for activated sludge process. Okay, you can use it for any biological process. Even if you know the kinetic parameter for anaerobic digestion process, you can use the same equation. You can plug the kinetic parameter and you can estimate what is the minimum SRT that you need in the system. Okay.
So design sludge residence time should be more than
Yeah.
what we have calculated.
Yeah, yeah. So yes, maybe design is minimum SRT could be 1 day, but you are not going to maintain 1 day. Maybe you are going to maintain 3 or 4 days, or maybe more than that. Okay. So now an example, okay, based on what we have learned, a completely mixed. So far any question? What do you think? But I think I was mentioning even yesterday. So here we kind of derive those equations for activated sludge process. But for any biological process, it is the same concept. You start with kinetic parameters, okay, kinetic equation. You do the mass balance in the system. Maybe if you work with other biological process, your mass balance going to be look little different, right? You are going to kind of come up with different form of equation. But end of the day, the fundamental concept is same. You need kinetic equation, you need kinetic parameter, you need process parameter. You kind of combine them, you come up with some design equation based on mass balance. Okay, so the fundamental concept is same for any biological process. So any questions so far before we kind of cover this example?
Here we have range of operational parameters. My opinion, I just want to tell you. If you have so much of data for different systems, if you consolidate and process the data, you can apply some machine learning techniques. You can get optimal operating
Yes.
process. Now, the machine learning techniques are highlighted in that.
Yeah, and I know now people want to use machine learning, a little bit of machine learning on everything. Okay, even we also use machine learning. Sometimes I collaborate with like the other researcher on how we can actually apply machine learning. One of the major challenge in machine learning is you need a big amount of data to train the model itself, right? So machine learning, end of the day, it's actually statistical model, right? So I don't want to offend anyone like who is working on machine learning, but end of the day it's like statistical model, right? But machine learning sounds better. Okay, so
And
but
one thing I would like to add here, it's a confusion among us that machine learning is going to predict something, but in fact it is not going to predict anything. It is guessing something
Again, we have to
because
go back
you have a data set, and within that data set only they are going to guess. They are not going to go beyond that data set. If it is going beyond, then only we can say it's a
Available
predictive
data
model,
and do its
but it is not.
inferences.
so for my personal perspective about machine learning,
Use an idea.
yeah, we should use machine learning for something that we cannot mathematically express. Okay, so let's say I have a complex. So I used machine learning. So we work on anaerobic digestion process. Okay, so one of my focus was I want to see antibiotic resistance gene. Okay, in digested sludge, because typically digested sludge is used for as a biofertilizer for land application. So now if we have a very high amount of antibiotic resistant, that ultimately going to come back, right? So we are going to grow food using those biofertilizer, right? And it's going to come back, and there are going to be a spread of antibiotic resistant, right? There is no mathematical model that I can actually use how different process parameter impact like antibiotic resistant in your digestate, right? I don't have any equation. So what I can do, I can collect some data, okay, I can train the model and I can guess, okay, if I have certain operating condition what could happen, right? But again you need big data. You actually train the model with limited amount of data, then it's not going to be, maybe you know the guess not going to be correct, right? So yes, it could be handy, okay, but for biological process I kind of feel like one of the major limitation is the amount of data we could have. And biological process, it's kind of like unique, like if you are running something, feedstock characteristics, wastewater characteristics, it may vary widely. So you develop some machine learning model, let's say you know, like so let's say you develop some model to see coliform concentration in effluent, like you know, one treatment plant here. You never know whether that's going to actually work for Delhi or some may actually wastewater treatment plant in South India, right? So it's really hard to tell, okay, but I think it's a very you know, kind of, it's an advanced tool. Yeah.
Go for practical feasibility. And even for COVID analysis also, X-ray will be US positive or negative. That was because of this machine learning techniques.
Yeah, machine learning again, like as I said, it's a handy tool, okay, but I kind of, my personal opinion, okay, I don't want to offend anyone, like you know, if you are working on or if you love machine learning, I also used machine learning in my research, but typically I prefer to use it for something, okay, that I cannot actually use, I don't have any mathematical equation, okay, or I don't have any mechanistic model, okay. But again, if you want to use machine learning, you have to compare with any mechanistic model if it's available, and then you can say machine learning, sometimes it can be handy tool for operator, okay. So you can develop in, like you have a model, okay, you can simply develop a software, okay. Sometimes operator, they don't care about mathematical equation, right? They can, let's say if you have a software, you plug the number, you see what happened. That could be a very good training tool for them, okay, or try to make some operational decision quickly. So instead of, like you know, doing some calculation, maybe model can quickly give some number, okay, or maybe model can quickly spit out some number and we can actually see whether it makes sense or not, okay. All right, so let's move on. A completely mixed activated sludge process with recycle, okay, SRT is 10 days, HRT is 8 hours, is used to treat primary effluent. Flow rate is 35,000 meter cube per day, and BOD5 concentration 193 milligram per liter. Estimate design safety factor, effluent BOD5 concentration, MLVSS concentration, sludge wastage rate, right? And we have that kinetic coefficient, okay. So I just put all the kinetic coefficient here. So first, design safety factor, we have an equation, right? So we can, based on the kinetic parameter and initial BOD concentration, we can estimate what should be my minimum SRT. So here minimum SRT is 0.25 days. But if you see in activated sludge process, the recommendation is from 3 to 14 days or something, right? Or maybe 3 to 10 days. So here SRT is 10 days. So 10 divided by 0.2. So I have safety factor of 40, right? So which is more than recommended, right? Yeah. So which is much more than recommended. Effluent BOD concentration is, and sometimes there could be a problem if you actually maintain really high SRT. So sometimes other, like you know, microbes, like some microbes might grow in the system that may not settle very easily, okay. That could be a problematic too. So too long SRT is not good as well. And effluent BOD concentration, we develop this equation, right, kinetic equation, where we can actually plug kinetic information and SRT, and we can calculate what would be the effluent BOD concentration. So my effluent BOD concentration is 1.25 milligram BOD5 per liter. MLVSS, we have another equation, right? We can put kinetic information, and SRT and HRT, right? So my X is 1188 milligram per liter, okay, which is slightly lower, okay, than what is recommended, but that one is TSS, MLSS. So when we say SS, it's suspended solid, total suspended solid, but when we say VSS, it's like volatile suspended solid. Okay. Sludge waste rate, we have this equation, right? So sludge waste rate, we know the volume of aeration tank, we can calculate from flow rate and HRT, so which is 11,550 meter cube, and now we know the concentration of biomass from here, right? We calculated the X, we know the V, and we know the SRT, so I can simply divide the mass of solid by SRT and calculate sludge wastage rate. Okay. So yeah, this is kind of like the end of topic 3. So what we have learned, okay, using kinetic coefficient and and kinetic equation and design parameter, like our process parameter, can be combined together through mass balance to develop some design equation, okay, that can allow us to predict some important parameter like, you know, minimum SRT. It can allow us to predict what would be the effluent concentration. It can allow us to estimate, okay, what is the minimum biomass concentration that I need in the system. Okay, so that's kind of like the implication of kinetic equation. Okay, so any question before I jump into the final topic for today? Yes.
Are there any ready-made packages for treatment of wastewater provided the input is given the expected output?
Yeah, there are actually many software right now.
Paid software.
Yeah, they are paid software. One we use.
Following those calculations, or they are doing on their own?
Well, first of all, you can develop some Excel spreadsheet if you want to. Okay, this is one thing. You can use commercial software. There are some software like, you know, BioWin, GPSX, Sumo. Okay, I know that Sumo is getting very popular these days. I think it was mainly developed in sorry.
Using Python also we can
Yeah, you can use any kind of coding, you know, like so, but the software that I am talking about, they have a like interface like software, like, you know, where you can calibrate the model. Okay, you can optimize the process.
Those softwares even layman can also design. A layman can also design instead of an engineer.
Yeah. So, but I can tell you, like in North America, we have 3 software, like, you know, very popular. BioWin, okay, this is actually developed in Canada. One company called EnviroSim. There is another software called Sumo. Sumo is more like open source software. Okay, so you can even modify that software. Sumo is getting very popular right now, and there is another software, GPSX. Okay, I personally used BioWin. Even like when I teach undergrad wastewater course, we actually kind of train students on BioWin software, which is kind of like they have I think educational version which is very cheap, but if you wanna use it for commercial use, which is one computer, you have to pay I think $5,000 or $6,000, okay, just for one key. And this is actually very popular in consulting companies in North America. But sometimes the input, the how you actually provide the input, it kind of vary in different software. Okay, so different software. And why I was, you remember that yesterday I was talking about readily biodegradable COD, slowly biodegradable COD, you know, like so every modeling software, they have the fractionation of input is different. They kind of ask you to provide the fractionation in a different way. So that's kind of like the calibration of those model. Sometimes software we kind of feel, okay, it's very handy, you provide some input, get some output. But one of the key challenge of using software in this field is calibration. Okay, so you have to do a lot of sampling, you have to do a lot of fractionation, okay, and you have to initially do some modeling and compare this with, like, field data, okay, and then you validate the model and then you use this model for any further optimization, okay. So that's, I think, the key challenge in using software in this field is the calibration, okay. Sometimes we kind of, yes, you can use default kinetic parameter, okay, but even one slide I was showing you some range, okay. So let's say you did lot of sampling from the field, okay, and you did not do any kinetic experiment, you just decided to go with default. Now let's say it's not predicting your effluent data. So now you don't know whether you did something wrong in your fractionation, or you did not provide maybe the default kinetic parameter, it's not well representative of what you have in the field, right. So you have to go back and forth many times and put lot of effort in kind of the calibration of the model that you develop, okay. So yes, like sometimes people kind of underestimate, okay, that, okay, what, software can give me everything, but actually it's not. You have to know how to effectively use the software or any kind of tool that you use, okay. Any other question before we jump into final topic? Actually, I have a final topic. It's not that long. What is time right now? 1:40. So we can, I think I'm gonna need about 25 minutes or so.
You can take maybe 5-10 minutes.
Now? Okay.
If you want.
Do you folks want break? 5 minutes break?
Also take break.
Huh?
You should also take break.
I can continue, I don't mind. You need break? Okay, if not, let's continue. So the very final topic for today is biological nutrient removal, okay. So what is biological nutrient removal? So although we focus a lot on carbon removal, right, or carbonaceous BOD removal from the process, we cannot overlook the significance of actually removing nutrients from the process. And the major nutrients that we have, inorganic nutrients that we have in the process is nitrogen and phosphorus. So what would happen if we don't remove nitrogen and phosphorus? Most of the time, majority of the wastewater, yes, please.
Earlier we were doing the biological process, sir.
Hmm?
Earlier we were using biological process.
Mm-hmm.
Do we need to add any supplemental nutrients?
No, typically we don't.
For example,
We consider that wastewater contain enough of nutrients that actually can be utilized by the microbes.
Microbial cells, some micronutrients are required.
You're talking about micronutrients like iron, manganese.
No, we typically don't. They are micronutrients, right? So they don't need a lot that you need to provide from outside.
Preparation of the growth media, some nutrients.
When you do laboratory experiment, yes, you prepare some media, right? But when you are talking about real wastewater, there could be some micronutrients should be enough actually for the
nutrient deficiency for the microbes, the growth rate also may affect.
But if you see, okay, now going back to your question. So what is the equation for, okay, let me clean the board first. We don't need them anymore. So we learned the chemical formula for microbes C5H7O2 N. So they don't need nitrogen that much. Actually, in your wastewater, you have much more nitrogen that they need for their growth.
That is the biomass.
Biomass, yeah.
It has to grow up. Another nitrogen is also required, no, sir.
Yeah, nitrogen is required, but wastewater has more than they required. They need actually more carbon, more hydrogen, more oxygen.
Sometimes suppose there is nitrogen deficiency, they add urea. Sometimes.
That's what I want to know.
And even sometimes if they feel that in influent they have low BOD, then they add glucose also. So that's why
Yeah.
the glucose will be
But
added.
in wastewater, typical domestic wastewater, you have like ammonia concentration 50 milligram per liter, which is enough.
In industry mostly they do this kind of.
Yeah, industry, if you are talking about industrial wastewater, yes, sometimes it could be. So, for example, industrial wastewater, sometimes BOD could be very low, that actually like you have to go with some other treatment process.
Normally, there is a practice of adding nitrogen source.
Urea in industry.
Even some biological nutrient removal process, you have to actually add carbon source there, right? So,
Carbon source
yeah.
itself is there in the
So now we can tell, okay, in wastewater we have organics. So why I am adding more organics,
Which is there,
right? Because it's not enough what we have.
nitrogen
So-
is there in the treatments.
Yeah.
So that's why if it is unknown wastewater, it's always better to do the characterization first. So what is the COD, what is BOD,
Yeah.
nitrogen level.
Yes.
Then you decide whether you need to add anything or not.
Exactly, yeah. So again, now let's go back to our topic. So major nutrients that we have in our wastewater is nitrogen and phosphorus. Okay. So and sorry, go back to this equation. So this equation you can see there is no phosphorus in there, but microbes, they still in their cell they need phosphorus, right? So it's too low that actually we don't actually include this in this chemical formula. Okay. But in some book you are gonna see there is P and then P is 0.0000 something, right? So it's very low. So now the nitrogen and phosphorus, most of the time wastewater treatment plant, after treatment they actually release the treated wastewater to surface water bodies, right? So and if we have high concentration of nitrogen and phosphorus, one of the major problem that we are going to have is eutrophication. So and then you can have summer algae bloom, which can actually lead to oxygen depletion in dissolved oxygen depletion in water bodies. So that can actually damage the aquatic ecosystem, right? And this is actually like the picture that I was mentioning yesterday from National Geographic. It's from Lake Erie, okay, in North America. One of the, I think it's the 4th largest lake in North America. And in 2011, in early 2000, they found actually every summer there is significant algae bloom, okay, in there, which was actually major problem, okay. Then after that actually people kind of identified, okay, what, like actually this is mainly caused by the nutrient release from wastewater. And then most of the provinces in Canada and states in US, they try to actually put a regulation for nitrogen removal, okay. So even 20 or 25 years ago, wastewater treatment plant, it was mainly focusing on BOD and suspended solid removal, but now everyone is also focusing on nutrient removal. And in the same treatment tank, they are trying to combine nutrient removal with carbon removal, okay. So biological nutrient removal process for nitrogen, you need two processes, okay, nitrification and denitrification. So in our incoming wastewater, nitrogen is actually coming in the form of ammonium, okay. So nitrification process converts ammonia nitrogen to nitrate, okay. Typically that's actually two-step process. First actually ammonia is converted to nitrite, ammonia to NO2, and then nitrite is converted to nitrate. So the first step is done by Nitrosomonas bacteria, okay, and the second step, nitrite to nitrate, is done by nitrobacteria, okay, and they are autotrophic bacteria, right, because they don't need organic carbon source. And nitrification process, it's an aerobic process because you want to oxidize ammonia to nitrate and then nitrite and then nitrite to nitrate, okay. So we can actually achieve nitrification process in activated sludge process. But what we need to do? We need to provide more oxygen, right? So most of the time we provide oxygen based on targeting carbon removal. But if we want complete nitrification, so ammonia conversion to nitrate, okay, we need to provide more oxygen, because some oxygen going to be utilized for oxidation of organics, right? But we need more oxygen to convert ammonia to nitrate. Okay, so one of the thing we need.
Ammonia level is more, right?
Yeah. So we need to. So if we want to combine both BOD removal and nitrification, what we need to do? We need to provide more DO or more oxygen in the process, and at the same time we need to extend the SRT. Why? Because nitrification, those nitrifying bacteria, they are very slow growing microbes. Okay, and in wastewater treatment plant, this is the slowest biological process, nitrification. Okay, and those microbes, they are very sensitive to temperature. At low temperature, actually, like nitrification is very slow. So at 20 degrees Celsius, recommended SRT is 4 to 7 days. Okay, but for at 10 degrees Celsius, especially for countries like Canada, that's a big problem. In winter, we can easily achieve nitrification, but in summer, in summer we can easily achieve actually nitrification, complete nitrification, but in winter that's a big problem. Okay, and nitrification is the first step, right? So if you don't have nitrification, the second step, it doesn't matter, right? First you have to convert ammonia to nitrate for your denitrification. So when temperature is less than 10 degrees Celsius, nitrification would be significantly reduced. So what some of the treatment plant in Canada they are doing, so they have aerobic zone. Okay, so in same treatment activated sludge process, okay, they have a big tank and they have different zone. Okay, some zone they have aeration, some zone they don't have any aeration. So denitrification process, you don't need any active aeration. So denitrification process convert nitrate to nitrogen gas, right? And so denitrification process is anoxic. You remember in the very first or second slide today I was mentioning. So they create different zone. One is anoxic zone. So this is for denitrification, and one is aerobic zone. So aerobic zone is for nitrification plus BOD removal. Okay. So in between they have another tank, okay, or another area. They call it swing zone. So what is swing zone? So swing zone, so in this tank you always have aeration, right? Anoxic, no aeration. But the swing zone, they have diffuser installed, but they run them only during winter. In summer, it's like more act like an anoxic zone. Okay.
Now I have a question. So first water should go to the aerobic zone, right? Because first nitrification should happen.
Yeah, but why we actually put anoxic zone first? Okay,
Yes,
this is actually to avoid. So what denitrification process need extra carbon source for those microbes. Yeah, they are heterotroph. They are not autotroph. But the problem is, if I have aerobic zone first, okay, aerobic process going to remove all the BOD that we have in our wastewater, right? So there are not going to be much BOD left, okay, for my denitrification process. So that's why it's actually better to have anoxic zone first, and then when you have recycled, nitrate going to come to this zone.
So through diffusion it will come.
Otherwise, if I put aerobic first and anoxic in the second stage, now not enough BOD. So that means I have to provide BOD. Like, as you are mentioning, you have to add glucose or you have to add ethanol in there, right? So, so that's why anoxic zone we put first. Okay.
It will come through the diffusion, right?
Sorry?
Diffusion, the nitrate.
Nitrate,
Means there is no barrier in between.
it's kind of like depending on the design. Like there are different tank. They are kind of, you create different zone in same tank.
Sequencing batch directly is the same tank.
Yeah.
So if different zone is there, then we have to recirculate water from aerobic zone.
So when actually even recirculate here, or there could be some internal recirculation. There could be some actually I have one diagram later on. You are going to see there is some internal recirculation in modified burden for process. Okay. So again, so in cold weather, what they do, sometimes they apply swing zone. So in summer, they stop the aeration because in summer nitrification is good enough. Okay. So they don't need a bigger tank. But in winter, when nitrification is slow, they kind of run aeration in there. So that means tank is bigger, more residence time, right? So kind of one way of dealing with the temperature. Now the second process, so after nitrification process can actually allow you to convert ammonia to nitrate, but nitrate you have to further convert into nitrogen gas for complete removal, right? And that process is called denitrification. So denitrification process, nitrate is reduced to nitrogen gas, okay, and denitrification process is done, we call it anoxic process. So that means chemically bound oxygen is used as electron acceptor, right? So here nitrate is my electron acceptor. So I need to provide electron donor, right? Because electron acceptor is nitrate and they are actually heterotrophic microbes. So that means I have to provide, there we call it actually facultative heterotroph, because facultative means they can actually kind of also, they kind of, so now for this process, okay, we need to provide, like sometimes depending on, like like we are mentioning that if we have anoxic process later, then sometimes you may need to provide like, carbon source from outside, okay. Sometimes the readily biodegradable organics, yes, if you put it as a first step, then maybe, the BOD in your, have in your wastewater, maybe it's enough for the denitrification. Then you don't need to add anymore from outside. But denitrification, the reaction, if you see, nitrate is finally converted to nitrogen gas, but there are many other actually intermediate compounds there. So you have nitrite, you have nitric oxide, we have nitrous oxide, right? So finally, so although we kind of see our final product is nitrogen gas, but many other intermediate compounds can be formed.
So nitrate is again getting converted into nitrite.
Yeah, sometimes, yeah, can be it can be converted back to nitrite. So now there are two options if we want to do denitrification in the same activated sludge process. So we can have pre-denitrification. So basically we have anoxic process first and then aerobic. So in that case we can have internal recirculation. We can, when we have nitrate produced here, we can actually recycle it back to the anoxic zone. Okay. So the benefit of this thing is those microbes can utilize the BOD that is coming from influent. Okay. So we don't need to provide anything from outside. But if we do post denitrification process, then we have to provide external carbon source. Okay. External carbon source here. So we could actually like add ethanol or acetate. Okay. But most of the treatment plant, instead of buying chemical, what they do, okay, in North America, most of the treatment plant, they are doing a process called sludge fermentation. So primary sludge, they do fermentation, okay, and then they produce different types of organic volatile fatty acid, okay, and then actually they separate those fatty acid, okay, and then they put it back in the anoxic zone, okay. So instead of buying chemical, they produce them on site to save cost. Okay, so you could have post or pre-denitrification.
I have seen the first one.
Okay, okay.
Second one,
Yeah, some treatment plant, like they use that. Like as I was mentioning, like even the treatment plant in Edmonton, the city where I live, actually their treatment plant is also using. They are producing sludge liquor. So organic acid on site and using that as a carbon source for their denitrification process. So now we talked about nitrogen. So what about phosphorus? Okay. So phosphorus can also be removed by a biological process called enhanced biological phosphorus removal process. So enhanced biological phosphorus removal process actually use some microbes called polyphosphate accumulating organisms. Okay. So those microbes, they can accumulate phosphate, okay, or phosphorus inside their cell, okay, more than they actually need. Okay. So typically in this equation you don't see any phosphorus there, but there are some microbes, they can accumulate phosphorus inside their cell, okay, and then you remove from the process as sludge. So when you remove the sludge, you are ultimately actually removing the phosphate, right, or phosphorus from the process. And EBPR process, it's a two-stage process. Okay, so the first step is anaerobic and second step is aerobic.
Instead of anoxic, here it's anaerobic.
Yeah, anaerobic. Yeah, and why anaerobic? So in the if you see anaerobic followed by aerobic, so what happened in the first step? So under anaerobic condition, okay, some of the BOD, okay, some of the organic, complex organic is fermented into simple VFA, okay, simple volatile fatty acid, and then microbes, those microbes actually consume that volatile fatty acid, okay, and then actually they produce a compound called PHB, polyhydroxybutyrate, okay, and that process, when they actually take the VFA, okay, produce PHB, okay, they release orthophosphate in the liquid, okay. So that process actually generate energy. And why we need anaerobic? Because anaerobic process can actually help you ferment some complex organic to simple volatile fatty acid, right? But when those actually they kind of go to the aerobic process, what happened? Now instead of producing PHB, microbes, they actually oxidize those PHB, complex biopolymer,
This
polyhydroxybutyrate. Okay.
hydroxybutyrate.
So instead of producing PHB, now they actually oxidize the PHB. Okay. So oxidize the PHB, and now instead of when they now they and that process actually release energy, right? And that energy actually utilized to actually accumulate phosphate inside their cell wall. Okay.
So basically in anaerobic process, phosphate is released and VFA is
Consumed and actually kind of stored like as a PHB.
In aerobic, that phosphate is consumed.
Yeah, yeah. So now if we look back for nitrification process,
Is there any name of bacteria?
so PAO, polyphosphate accumulating microorganisms, polyphosphate accumulating.
We missed for denitrification, Azotobacter.
They are facultative heterotrophic microorganisms, or we call it denitrifying bacteria. Yeah, yeah. So nitrification process is aerobic or anaerobic? Aerobic, right? Aerobic. Denitrification,
Anoxic.
anoxic. How about enhanced biological phosphorus removal, EBPR?
First anaerobic.
First anaerobic followed by aerobic. Followed by aerobic. And our BOD removal is aerobic, right? So you see, if I want both carbon and nitrogen and phosphorus removal, I need aerobic, I need anaerobic, I need anoxic, right? So we can, like, so now instead of one single tank, yes, we can still have one single tank, but we can create mode zone. Okay, we can create mode zone. Okay, one thing can be anaerobic, one thing can be anoxic, one thing can be aerobic. Okay, so that's how we can actually design some system that can actually integrate both carbon and nitrogen removal. Sorry, carbon and nutrient removal. Okay, so both carbon, nitrogen and phosphorus. And how we can achieve that? One example, there is actually process. Oh, this is actually I think it's a transmission electron microscope. Okay, imaging. It's kind of showing that how after bacteria they accumulate the phosphorus inside their cell, like how it looks like. Okay.
So can we remove the nitrogen and phosphorus at the same time? At the same time, because anoxic and anaerobic is kind of similar.
So anoxic and anaerobic kind of similar means
or we can create two zones.
Yeah, that's actually what I was going to show right now. Okay, so how we can actually integrate all process in one single tank. Okay, so we can actually create mode zone, and that process is actually called modified Barden-4 process. So this is actually, you have 5 different stages in a single tank.
What is the name?
Modified Barden-4 process. Okay, so it can actually allow us simultaneous nitrogen and phosphorus removal with carbon removal. So we have in one single tank, we have anaerobic, anoxic and aerobic zone.
This is same like sequencing batch reactor?
They are they claim that, okay, you know what, typically it's not sequencing batch, it's kind of like in the same tank or same process, they have different zone. They kind of divide in different zone.
Actually, we converted one ASP process like this.
Okay.
In Hyderabad.
Okay.
So already there was one existing plant of 30 MLD. It
Yeah.
was having ASP process. So space constraint was also there. So we just added this anaerobic and anoxic process.
Yeah, like most of the treatment plant,
In the aerobic chamber, we created zones, just like anoxic zone.
Yeah, exactly. Like most of the treatment plant, for example, most of the treatment plant in Canada, like especially in the city of Toronto, they were built in 1950s. Okay, and in I think between 2010 and 2015, they started to actually upgrade. And at that time, they are actually incorporating all of this. So when I was a PhD student, I actually did an internship with them, like with City of Toronto for about 6 months. And at that time, I was the one actually who was kind of modeling with some consulting companies and see like how that process is going to work, because that was kind of new for them, like creating different zone. They put a lot of sensor there at that time. So yeah, like when you can modify the existing system by creating different zone. So why? So you can see we have anaerobic, we have anoxic, then aerobic, then anoxic, then aerobic. So why we put anaerobic first? Any thought?
Pardon? I didn't.
So why we put anaerobic first?
Because first we have to remove the phosphate.
First we have to remove the phosphate, and so anaerobic process typically need more BOD, right?
Yes.
So it's better to put that as a first stage. Then anoxic. Why I don't put aerobic there?
Because
Even anoxic process also need BOD, right? So that's why anoxic, and then aerobic, and then you can see there is an internal recirculation. So we can recycle some nitrate back to the anoxic process for denitrification, right? And then finally, to make the process more efficient, we have more anoxic and then more aerobic. So In single process, we can combine carbon, nitrogen, and phosphorus removal. Okay.
Just curiosity, when this process was designed, I mean invented, modified,
Honestly speaking, I can't recall like right now.
but approximately how many years back?
Maybe we can Google. Yeah, if I don't have, if I don't know, I think it's better not to answer. Yeah. So any other question? So I think here we end for today's session. Okay. So
What is the major source of these fluids?
major source of
Nitrogen, phosphorus, sulfur.
nitrogen is mainly coming from urine. Okay.
Source domestic
Industrial, actually, it's a kind of specific to industry. So what industry you are talking about?
I think municipal wastewater also, that smell, ammonia smell.
Yeah, it's mainly coming from urine, right? It's mainly coming from urine and protein degradation. Like we eat a lot of protein, right? So ultimately, it's gonna be the wastewater, right? But depending on industry, it might vary, right? But typically, ammonia concentration in domestic wastewater, more or less, I think it doesn't matter which country you go, it's close to 50 milligram per liter all the time. Yeah. Any other question? Yes.
Sir, why algal growth is a problem? It is technically reducing nitrogen and phosphorus content. All we have to do is control the algal growth.
The problem is it covered the surface of the lake, right?
Dead biomass
Yeah.
is a problem.
Same question. Sir, eutrophication is always a challenge, or it can be a solution for anything?
So, well, LG, we know that actually we can there are even LG based wastewater treatment, right? We can actually grow LG to remove nutrients, and then we can actually use LG biomass to produce something else more valuable, right? Or we can for lipid extraction process, lot of different other process actually. But when you have that in, let's say, surface water bodies like lake or river, then it's a problem for your aquatic ecosystem, because you are covering the surface, there are going to be oxygen depletion. Some of those, like aquatic organisms and plants, they need the sunlight, right? They are not going to get it, right? So you are basically disturb the ecosystem. But yes, if you take it outside, do it in engineered based system, grow LG, then that's a different thing, right?
So in this continuation in eutrophication, when the algae die basically,
Yeah.
so then the decomposers comes into the picture, start eating those dead biomass and consume oxygen, the dissolved oxygen. It's become very, very less and critical to survive all other aquatic organisms. So that's why eutrophication is a big
Wastewater treatment plant with algae.
So that's the control. You are growing in a control.
So you are doing it in a like in engineered system, right? You are not disturbing like ecosystem in a river.
Only algae. You are growing only algae. There are no other.
And it's not just about algae. Like sometimes some cyanobacteria can actually grow, which is actually toxic. Any other question? Well, if not, so here we end today's session, I think, and I think you have a field trip today, very exciting field trip. So, and tomorrow we are going to cover, we are going to move from wastewater to waste. Okay, so we are going to talk more about solid waste, and tomorrow another two important topics that we are going to cover are going to be anaerobic digestion and composting. Okay.
So somewhere I have seen removal of the dissolved oxygen from wastewater was an object. What for it?
Sorry, what?
Removal of dissolved oxygen in wastewater.
Removal of dissolved oxygen from wastewater.
There are some experiments I have gone through. What is the objective for that? I don't know. What is the
So
removal of dissolved oxygen from wastewater? So why do we want to
It's a research paper or what?
It's a research paper.
Dissolved oxygen
Using
or
enzymes you have
related to BOD?
I have no idea. Or maybe BOD, not dissolved
No,
oxygen.
not dissolved. Dissolved oxygen is
Result as
required.
people have done, sir. I've gone to some articles.
Hmm.
Main concept is DOD only, sir. That main DOD will come to the DO only. So main concept is
Maybe you can share the article.
Yeah, maybe you can share the article tomorrow.
DOD तो
Ah, exactly. So generally the DO is it's related to BOD only. Inversely proportional.
Enzymatic reactions. It was an enzymatic reaction.
Yeah, there are actually like thousands of different kind of bioprocesses. So it's hard to tell which word you are referring to, right? So
Why we are here doing 2 times anoxic aerobic, anoxic aerobic? That I could not understand.
huh?
Here,
Yeah.
why 2 times we are repeating the?
So it's kind of like the process, like instead of multiple. So if there is anything remaining, it's gonna be actually removed in the second stage. Yeah.
So then you
So
don't
it
need
is
an
kind
internal
of
circuit.
trying
Yeah.
to improve the efficiency.
Hmm?
Trying to improve the
Trying to
efficiency
improve the efficiency. Yes.
by adding the tanks in CV.
Yeah. Secondary sludge to the anaerobic tank.
RAS. Return.
So when it's showing this, it's kind of showing in the single tank. Okay. So single tank with different zone.
No, but here we have the aerobic microbes.
Yeah, aerobic microbes. Yes.
But why we are circulating that microbes to the anaerobic tank?
No, that is what he is saying. It's not a separate tank. It's the same tank. It's in a same tank with a different zone.
Or maybe it's not
And
anaerobic.
again, you have to actually maintain certain SRT for this, right? You have to maintain certain biomass concentration in there as well.
Or maybe it is not going to the anaerobic. It is just you have the system boundary, and it is going to anoxic tank.
My understanding, it's single tank with different zone. Okay. Single tank with different zone. Yeah, I cannot really answer because this is a process developed by something. Maybe if I develop something, I might have a different opinion. Yeah,
Find the answer.
yeah. All right. So thanks everyone. See you tomorrow. Okay.
Okay, and
Yeah.
so we'll end the session now and we can proceed for the lunch. So after that, the bus will arrive here only for the industry visit, and the bus will drop off here. But I don't think someone from the Lava city is coming. They want to get down, returning instead of coming back to the campus again. I think you can get down at your location. Although the bus route will not change for you.